Um, this is an important meeting, and I must congratulate ILSI for organizing it at a very right opportunity and time. And with all those who matter, I could see they're all here, and we have been, in fact, getting a lot of benefits from the presentations. We missed it yesterday, but today I was quite happy to see the progress made, kind of research which is being undertaken, to have ameliorating effect or impact of climate change. And what we are going to do, in fact, are doing in agriculture, and that to Indian Council of Agricultural Research. More, of course, Professor Datta will be there to talk about it. But then I come from one part of agriculture, and that is managing the genetic resources. And when we talk of various adaptive strategies, imperatives we have in front of us, what is more important and which has not been harvested or harnessed so far, to really, reduce the impact of, climate change on agriculture is something that is crop genetic diversity. When on one hand we have this diversity, on the other hand we have challenges of climate change, can we bring together and try to really see and move forward? And so in next about 20 minutes, I'm going to be more specific to a particular crop, but I'm just going to give you that as a case study, which could be utilized for any other crop for that matter, where we have all these kind of diverse genetic material available in India, as a resource, as a big resource. I don't have to go into the details of it. There are many challenges facing agriculture. You all know it. And also you could see exactly the same message from Mark Rosgrand, presentation as well, climate change followed by shrinking land and water as natural resources, and all others, also very well has been already covered. But when we talk of the impact, most of the climate change studies have shown that significant decrease in crop yields in South Asia, which includes India, of course, has also been talked about clearly in several of these slides presented by Mark Rosgrand. But I'm specifically, in fact, making a case here for wheat. For food security in India, wheat is of prime concern. No doubt rice is important. Many other oilseed, pulses, vegetable fruits are all important. But when we talk of food per se, I think wheat is a major crop. And therefore, I'm going to talk about it wheat because as someone talked about C3, Geeta talked about it, it's a C3 is going to be, impacted again because of the temperature more, if not because of high CO2 more. But then again, when you see the different growing zones of in India, almost all out of these 6, you can see they're all getting affected badly, not only in the future scenario point of view, even currently with regard to the effect on crop by heat stress. In some cases, it is heat stress throughout the plant growth, as you see in the central zone. In some of the cases, you can see terminal heat stress, as you can see northwest plain zone on the northwest plain zone, as you can see here, which is terminal heat stress. But again, you can see in the central zone, it is heat stress throughout. In all growing zones, what I'm trying to say is that there is a impact of climate change on wheat. And to what kind of an extent is there climate change? Increase in 1 to 3 degrees Celsius temperature may reduce yield up to 10%. That's predicted by 2020, which is around the corner, which is already there, in fact, if you talk about the way the agriculture is progressing. And then 5 to 7% decline in wheat yield for every degree rise in temperature is expected. And therefore, we need to have these adaptation and mitigation strategies. And many of these, first of these is development of heat-tolerant varieties, or changes in crop management practices, including use of water-saving technologies, which is also one of the topics in this particular seminar and is being talked about it, and weather forecasting and risk management measures as well, as has been mentioned by other speakers. I'm going to mostly talk about it, whether development of heat-tolerant varieties is possible or are there, or what strategy should we be using in mitigating the effect of adverse effect of climate change. So therefore, when we talk of major constraints for wheat productivity, which I'm making as a case in case of wheat, I said before, is a heat stress, followed by shrinking water and land resources. But then these are physical factors rather, physical environmental factors, heat stress, shrinking water, land resources, etc., which is not entirely in your hands. But what is in our hands is breeding. Can we do better breeding to have those improved cultivars? Even in breeding, there are constraints, therefore. What is that? Lack of screening and phenotyping methodologies. Scientifically speaking, you would like to do the breeding, you have probably the genetic resources, but then lack of breeding effort, pre-breeding efforts is one of them, and narrow genetic base. If you look into this very clearly, not only for wheat, you will find that true for most of the crops in agriculture. Not only in India, it is a global scenario today. I have seen in some of the countries where we have visited, some of the gene banks, this turns out to be very true, and this is so also the exercise we have done with the help of breeders. So therefore, ultimately, the important message here is that we need to use this twin pillar strategy. And what is that? One is germplasm evaluation for widening genetic base. Because for any resistance development, resistance against not only biotic stresses, against abiotic stresses as well, this is very important that we do and go for genetic widening of the base, and then take up pre-breeding and development of, therefore, finally, climate resilient varieties. So this is a message which I'm trying to give you here before, is that utilization of genetic diversity. And when you talk to, when you talk about India, India is a gene-rich region. We are so fortunate, in this part of the world that we are one of the 12 mega biodiversity centers and one of the 17 mega diversity nations. And I'm not going to go into the details of it, but not only in plants, even in others, animal, fish, or microbes and insects, we're all quite diversity rich. And this becomes our natural biological resource for adapting ourselves to a change in climate or to other, climate, vagaries of weather. And when we talk of our biological resources in India, particularly focusing on plant genetic resources, we are the third richest or third largest gene bank in the world. When we talk of our total number of holdings, you can see here in the yellow, there is about 0.4 million, 4 lakhs, 0.4 million total number of accessions with very good resources in case of cereals, millets, forages, pseudo-cereals, grain legumes, oilseeds, fiber crops, all related with not only for food security, even for fodder, for the animals, the cattle, which is very important again in India. And we are very rich and we are very fortunate. But the question is, are we utilizing it? Are we making use of it in fighting, like changes of climate or any other issue which we are currently facing at the moment? But then, are we doing it or not? Anyway, we thought our message and our goal, we set it about, 3-4 years ago, that we need to enhance agriculture production by enhanced utilization of PGR. Not only conserving them as an ex situ gene bank which we have, rest of the world has it. But the question is, I'm putting it before and before, are we making value out of it, or are we making use of it? Our breeders using it, to broaden the genetic base and again to prepare themselves for fighting the climate change, particularly as the message is today, we're trying to do it. But then if you go back into this slide here, you will see that we are dealing with, in fact, this number 0.4 million belongs to about 1,500 species, crop species, which are all for food and agriculture, But then when we talked of and we set a goal ourselves for increasing the production by enhanced utilization, we couldn't have done justice with 1,500 species. We decided, therefore, to prioritize. And these prioritized crops are 15 here, as you can see, which contributes to 60% of the total, gene bank accessions we have in our germplasm collection out of 0.4 million. 60% belongs to these 15 crops. And these 15, I'm sure all of you would agree, for not only Indian food security point of view, global food security, nutrition security depends on these almost, most of the cases, I would say, barring few. I don't have banana. Banana is, of course, here again. Potato probably I don't have, in this list. But then this is what is the message that, and we try to evaluate this germplasm against all these kinds of factors listed here, be it resistance to biotic stresses, resistance to abiotic stresses, nutrition quality, we cannot, again, leave it behind, then earliness and photo and thermo insensitivity. These are some of the factors which will automatically bring in, adaptation to climate change. If you have some early flowering, it will escape those. So there are different mechanisms. Not only you have to have resistance and tolerance, even earliness, you can escape, or avoidance mechanisms can also be utilized when we talk of improving these crop plants. But then again, before I took up this study, we prioritized it, but then I went around all the breeders in the country, in India, and did this analysis. And we found the gaps. Again, the same thing which I mentioned before, I'm repeating it, narrow genetic base of the breeding material. And that's because of certain reasons. I don't want to go into the details of it, which is listed here. But the next is we have a lack of long-term strategy for broadening the genetic base, also not existing at the moment. But now we have prepared a kind of a platform, which is there in the 12th plan, more Professor Datta's guidance, we have done it. And then poor characterization and evaluation state of the germplasm, also it turns out. The point I was trying to make you is this, that we have this gene bank, breeders are not using it. Also, there's a constraint that we do not know what kind of material we have, what value it has. So we need to evaluate it also. Therefore, what we did, we wanted to evaluate to harness this power of germplasm. And that is when we talk of adaptation to climate change, there is a potential here, particularly this is the case of wheat and rice. When you see the potential yield or, compared with the climate, with attainable yield and measurable yield, there's a lot of potential. And here one can use the same slide for, harnessing the potential of any technology for that matter, But here I want to fill this gap between potential yield and the measurable yield or attainable yield by utilizing the power of available germplasm. Because ultimately the farmers would, of course, depend on many of the cultural practices. But important is the best technology available to the farmer could be only the seed, which itself has got all that kind of inbuilt, resistance or quality or vigor, which is required to fight, against many of these climatic factors. So therefore, we thought we can try to utilize the genetic resources. And also I would like to inform you that what all genetic resources we have, I gave you a whole list, but when we talking of genetic resources, let's say in a country like India, it is from all other corners of the world. Here you can see, briefly I'm mentioning to you here, wheat which we have collected from exotic sources, which are conserved from many of these continents. You can see all the continents, Asia, Africa, Australia, Europe, North America or South America. And also within India, we have from all those different 6 zones which I mentioned where wheat is growing. So this total number of germplasm, we call it, or different lines of wheat, we call it, are about 20 and 25, in between 20, 25,000 in our gene bank. Now, if I have to use them or develop a variety, resilient to climate, I must try to make use of it. So therefore, what we did, we took an initiative to characterize and evaluate the entire set of wheat. That's about 22,000 accessions of wheat, 22,000 accessions of wheat, and we identified trait-based reference collections. Trait-based, I mean the one which are tolerant to heat stress, and then we try to share with the breeders, and breeders will make use of it in wheat improvement. And this is not happening at the moment, so therefore this initiative, and if not is happening, is not happening utilizing the power of entire germplasm collection which we have, and therefore we initiated this activity for evaluating entire set I'm talking about here is 22,000 wheat accessions, at a single location, and then that means again you have to move to different locations if you're talking of, under different climatic conditions. So we started with this first in one of the best forms of HAU in the northwestern, India, about 150 kilometers from Delhi, in northwest part, where there is a university. We did that there, and we, in fact, there, after we were successful in that single site with all 22,000 accessions, we went on to do the phenotyping for climate resilient or heat tolerance as well in one of the, as you can see here in the red, for terminal heat stress. And then we wanted to do the evaluation at one of the best locations. Then also we wanted to evaluate the entire germplasm against various biotic stresses, particularly against resistance to multiple rust diseases, like brown, yellow rust, stripe rust, leaf rust, which are so very important, to be studied. And there is a pressure again of climate change again for the different strains getting, different races, getting evolved over the time. So we would like to prepare ourselves whether we have that material which can withstand, this kind of pressure as well of the changing races and strains of, various, fungi or even bacterial blight for that matter in case of wheat. So finally, we prepared this kind of a roadmap at 3 different locations for the 3 different traits, or 2 different traits rather, terminal heat tolerance and biotic stresses, and we were finally successful, and I'll share with you quickly those results. These are some of the locations I'm showing you in India where these experiments were carried out. You can see here very clearly terminal heat stress tolerance near Delhi. This is in Issapur Farm, and then in Hisar, this is it. And then for yellow rust resistance in Punjab, this is another state, and then for late blight in Cooch Behar and West Bengal, these are different locations, and this was all experiments were done from 2011, 12, 13, and now 13, 14, we are progressing with it. And we are progressing with entire 22,000 accessions, which is not easy, by the way, my dear friends. I'm just sharing with you. This is the experiment. I'm very happy that it's been done for the first time of such a mega scale in such a scientific and designed experiment, statistically speaking. And this is what the entire field show I'm showing you here, field layout. You can see we're in about 20,000 accessions with various checks, just giving you a view. And this is the genetic variability, as breeders tell me, at its best. Because those breeders, if they're here, would appreciate, if at all, any breeder would have seen a genetic variability, would have seen 500 lines or 1,000 lines or 2 or 5,000 lines or 10,000 lines, but never 22,000 lines at a single location, 3 lines each, so that they can really go there and select and then prepare themselves for the future and even for the present food security point of view. And this is what is, again, specifically I'm showing you for terminal heat tolerance. The trial conducted 2 different dates of sowing as we normally do it, so that there is an effect of climate change, truly speaking. And we also, these are the details, I don't want to go into it, what were those 22,000 accessions. But then important is, I just wanted to show you the experiment when it was carried out, whether there was an effect of high temperature, physically speaking. So we had 2 different dates of sowing. This is the anthesis date in the normal zone, and this is the anthesis in the late zone. You can see here in the late zone, I mean, the experiment, whether maximum temperature or minimum temperature. In either case, you can see that they exposed during this particular period, which is the grain growth period. They were all exposed, as you can see here, 31 degrees in case of normal growth, normal zone, and 37.5 degrees. So they were exposed to, I mean, this is the normal scenario in the farmer's field, by the way, which we followed. And this is just to show you, some of the friends may like to see this. There is a kind of variability. I don't want to go into the details of it, but we could see something which we had never seen before. This kind of variability in wheat, you can see again the leaf angle or the waxiness, on the leaf, etc., which are very important, for terminal heat tolerance. In fact, one of my friends, Dr. Sesha, is here from ISRO, and they also had done, using spectral radiometry, all those, phenotyping which is required, and probably sometimes, he'll be able to share with you that information. But important is canopy temperature depression, in relation to ambient temperature, whether there is a canopy, whether the plant is able to maintain a lower temperature compared to ambient. Whether there are genotypes. Here we could find genotypes which were able to lower down the temperature of the canopy or the leaf, I'm talking of the foliage, compared to the ambient, to up to 12 degrees Celsius, which is wonderful, up to 12 degrees Celsius. That means they have that mechanism which could be then transferred to some of our high yielding, but otherwise susceptible to high temperature tolerance traits. And this is, finally, we were able to identify, this is out of those 22,000 accessions I'm showing you, out of 22,000 lines, 3 lines only I'm showing you where the grain yield point of view, the heat susceptibility, they were highly resistant. Zero was the susceptibility index. I mean, or they were highly tolerant. But whereas, the percent reduction in grain weight was also, you can see here, percent reduction was only 8% in some of these lines I'm showing you. So these are kind of information material which we never could have gathered unless we had done this kind of exercise. So finally, we have this, as we call it, as reference set for terminal heat stress tolerance, with about 3,000 lines, which has been further narrowed down now to 300, and which can be now shared with the plant breeders or with the molecular breeders for any kind of molecular analysis. And similarly, we have, of course, we are further doing this experiment, in 2012-13 we did it, now we are doing it in 13-14 as well, with those 300 lines. And similarly, we identified in the yellow kind of bar you will see row that about 5,000 lines we were able to select out of 22,000 resistance to multiple stresses, stem, leaf, stripe, etc. Now this number has come down too after the analysis we did it further with the help of plant pathologists throughout the country. This experiment not done by one organization, by several organizations, and 50 to 100 partners, or scientists and workers involved in it. And we are now narrowed down it to 660, which is further, now being tested against different races in Shimla, one of the centers we have where they have got all these strains and races, collected with them right from 1930. And therefore, we have finally this reference set for biotic stress tolerance, means against all resistance to, various rusts. But then once we have this reference set available, which could be directly used as a parent as a donor by the breeders, but we have available with the genomic tools, we can even go and further association analysis, identifying QTLs, etc., can also be, so that material is now available for the molecular breeders to, study the association analysis using genome-wide association or candidate gene approach, and one can do it now. And there's a roadmap. Finally, see, we have got this characterization for wheat, for different traits, terminal heat tolerance. Also, we have now georeference to wheat. That's another technology which we are using, and which I'll just discuss with you briefly. So this is the work of the entire team I'm mentioning here. And one more thing I thought I'll share with you. This material in our gene bank would have been stored for the last 50 years. We would have collected it from different sources. Now, whether that was viable. This was the first time that we took out this kind of material. Here, we were so happy to see more than 90% of the germination. So that shows our conditions of storing them in minus 18 to minus 20 degrees Celsius, one of the third largest gene bank I mentioned before. There is a viability of that material could also be tested. The material could be replaced now back into the gene bank with that kind of power we have. And of course, this I'm very happy to share with you. This one particular was awarded as well. But this is another technology which we're using it. Based on our GIS tools, georeferencing the collections which we would have done it from various locations in India, as shown here on the Indian map. For wheat alone, we are doing it for 6 different crops, 5 different crops including wheat, and then accordingly we are preparing our plan for collecting them now, according to the climate change scenario, or using those varieties. This is entirely done with the varieties. There are about 350 varieties we have in case of wheat. So using those varieties, the varieties which are adaptable today to this particular scenario, and can be now utilized for the future scenario. And also there is a potential of underutilized crops as a wonderful reporter of genes, which can thrive under very harsh conditions. And we can, not only this, they are entirely, useful for nutritional point of view as well. So this is another, I think, kind of genetic resource. We are talking about it where we are quite rich, and we hardly otherwise talk about it. But since this is an important seminar, I thought we can be a wonderful source for resistance to climate change. And we are, of course, utilizing all the germplasm, not only in India, but we are trying to increase South-South cooperation, where an organization like ILSI could also be otherwise, important, via media, to collaborate more. And we did this recently, last year, a global consultation, this year, in fact, early this year, a global consultation, use and management of agrobiodiversity for sustainable food security, but with a focus again on climate change and nutrition is very, very important, and we are preparing a roadmap for this. So with this, I thank all those who matter, right from the friends in ICR, particularly Professor S.K. Datta, the Deputy Director General Crop Sciences, Dr. Sandhu, who was that time ADG Seed, and many others, as you can see here, directors and vice chancellors, and all those partners from even CIMMYT and ICARDA, and it was a teamwork, and I thank you very much for your patient listening, please. Thank you very much.